We're going to continue talking about this molecule called RNA, or ribonucleic acid, and its role in this whole process of making proteins. So in the last lecture, when I started talking about this process that we call protein synthesis, we're talking about how does DNA actually get used, right? So we have this cookbook of all the proteins that could ever be produced by a living organism. That is what DNA is. So now we have this issue of, all right, so the DNA is in the nucleus, but the proteins need to get made outside of the nucleus, so how do we solve that problem? And we talked about in the last lecture this molecule called RNA, or ribonucleic acid. And RNA has a very important job. Its job is to copy the information from the DNA and bring it to the ribosomes where it can actually make the protein. So we're going to talk a little bit more about that in today's lecture. So let's get started. So we use this term genetic code, and the genetic code is this recipe that I keep talking about. And what the genetic code allows for is it allows for almost an infinite number of proteins to be made. And I'm going to give you an analogy that I think you're going to understand. How many letters are in the alphabet? Well, the answer to that question, if you think about it, is 26, right? So there's only 26 letters in the alphabet. But how many words are there? There's a limitless number of words. You can come up with as many words as you want. So how do you make a new word? Well, you just take the letters and you rearrange them, right? So for example, if I took the word, and I talked about this in the last lecture, dog, and I rearranged those letters and it was God, well, it's the same letters, but it's a different order. So now that word has a much different meaning. Well, proteins work the same way. There are 20 amino acids that exist in, as far as we know at this point, right? There's 20 amino acids. And a protein is made out of amino acids. That's what it is. It's a long chain of amino acids that make a shape. And then that shape is going to determine what its job is. So the genetic code allows for an infinite number of different proteins because all you need are the bases in a specific order to make a certain protein. But what we're going to talk about today is how does it actually get read. So every 3 bases of DNA or RNA is called a codon. So a codon is the code that codes for a specific amino acid. So every amino acid has a specific code that codes for it. For example, GCA is what codes for the amino acid alanine. So GCA codes for one of the amino acids called alanine, and we're going to learn how to do that in this section. So let me just give you a little bit of a different example to explain it. Okay, so what I have here is I have a very short statement, right? If you take a look at this, you have a bunch of letters all put together. Now, if I take a look here, what I notice though is they are not just letters, they mean something. There's, I could write a bunch of nonsense, but if I take a look here and I start to really take a look at it, what I notice is every 3 letters, I can draw a line, right? And each of those 3 letters means something. The car was red. The means something because that means it's the beginning of the sentence. Car, we all know what a car is, right? We got a little car here. We got our car, so we know what a car is. Was means that it already happened, so that has a meaning. And then red is the color red. So if I take a look at this, if I pulled out these, if I remove these little lines I drew, I just see a bunch of letters that are put together. But our brain knows how to look for words. So our brain has identified that those letters have meaning and they're words, and we put lines in there. You automatically put those lines in without thinking about it. You knew, if you just looked at that, you saw the car was red. Well, that is exactly how a codon works. It's every 3 letters. So if I take a look here, what I find is I have a bunch of nucleic acids all lined up. I'm sorry, a bunch of nucleotides. So if I put breaks in there every 3, what I find here, I have 4 separate codons. Now this G is just an extra letter here, and we'll talk more about that later. So each one of these would code for a specific amino acid. So how many amino acids would be in this protein? There would be 4 amino acids, and those amino acids would pair up with each other and make the protein. So the type of protein is going to be determined by these 3-letter codes called codons. So we just talked about this idea of a codon, which is that 3 base pair code. So if you have a strand of 3 billion base pairs, that means there's a billion codons in there, right? Because you take 3 billion divided by 3, you get a billion. So our DNA strand is read every 3 base pairs. That's how we read it, and that's how we do this. Now, if let's say I want to actually make a protein, so I want to read that DNA and I want to make a protein out of it. Well, that happens outside of the nucleus. And the reason why it happens outside of the nucleus is, for one thing, because proteins would be too big to get out of the nucleus, but also that's where that's where the proteins need to be. You're building them outside of the nucleus because they're going to get used outside of the nucleus. It would be like if I had a car factory and I built the car in the office instead of in the factory floor. You can't get the car out of the office. So we have these things called RNA, and there's actually 3 types of RNA, and that's what I'm going to finish up with today with this lecture. I want to talk about the 3 different types of RNA and what they do. The first type of RNA is called mRNA, or messenger RNA, and that's the one that I've been talking about up to this point. So the mRNA is essentially, this is the RNA that copies the information from the DNA. So the mRNA is the only one that's going to be inside the nucleus. The mRNA is produced in the nucleus, and it's done by reading the DNA, and it copies the DNA. So the mRNA is very simply a messenger. So if I were to give an analogy, let's say I work, in the building where we work in Methacton. Let's say I have the plans on how to make a piece of artwork. I want to make this artwork, but I don't have any art tools. I don't have any paint. I don't have anything else. So I might draw a little sketch of it and say, hey, I want this to be made. And I might give it to one of my students to carry down to Mr. Coons' room. And then Mr. Coons will look at it, and then he'll produce the art. So think of the messenger as the person who takes the information from the plans to where it actually gets made. So the messenger RNA copies the DNA in the nucleus, and it brings it out of the nucleus to the cytoplasm, and it attaches to the ribosomes. So it carries the instructions to the ribosomes on how to make a specific protein. So mRNA is the messenger RNA. The second type of RNA is called the rRNA, or ribosomal RNA. And the ribosomal RNA is very simply the RNA that makes the ribosome. So what is a ribosome made out of? It's actually made out of RNA. It's made of a special type of RNA that wraps around, and it actually makes a shape. And we'll talk more about it later, but there's 2 parts to it, and it actually attaches to this strand of mRNA. So the rRNA, or ribosomal RNA, is actually what makes up the ribosomes, and that is what determines how to make the protein. So the rRNA actually makes the proteins, and it reads the RNA, the mRNA, and it assembles the proteins. And the last type of RNA is one that's going to be hard for me to explain right now until we get to the actual process, but there's a third type of RNA that is called transfer RNA. And transfer RNA has a very important job. Transfer RNA's job is they carry the amino acids from the cell, the cytoplasm, to the ribosome, so that way you can make the proteins. So think about a scenario where the ribosome cannot move. The ribosome is stuck to the mRNA, but it needs amino acids, because what is a protein? It's a bunch of amino acids. So I'm going to give you an analogy of a cook. So if you've ever seen a restaurant before, there's a lot of times in the kitchen there's a chef, and the chef's job is to put all the food together. But if it's a busy restaurant, the chef needs help. So the chef probably has some helpers in there. So let's say the chef is making pasta, right? And has this really complex recipe in front of them. And the chef reads the recipe and needs to put some stuff together. The chef might say, hey, you, go get me some eggs, right? And hey, you, go get me some flour. Hey, you over there, go get me some tomatoes. And those people that are going to help, they're going to go out and get the ingredients and bring them back to the chef. Those people are like the T or the transfer RNA. Their job is they go and they pick up the amino acids and they bring them back to the ribosome, so that way the ribosome can put together this protein by combining these different amino acids together. And that's essentially the 3 different types. So if you take a look here, this is a picture of the 3 different types of amino acids. I'm sorry, the 3 different types of RNA. This is messenger RNA. It's single-stranded, and its job is to copy the DNA and bring it to the ribosome. Over here we have our ribosomal RNA. So this is the actual ribosome itself. So this mRNA would travel to the ribosome, and the ribosome would read it like it was reading a code, and it would travel through the ribosome. And then the tRNA is this weird looking guy right here, and the tRNA, its job is it goes and it picks up an amino acid and brings it back to the ribosome so the ribosome can put it together. So I'm just going to show you a quick animation of how those 3 work together. So I'm just going to give you a quick rundown of these three types of RNA and what their jobs are. So the first job would be the mRNA. So this is the mRNA here, and it's going to go in and it's going to copy the DNA. And I'm going to explain how it does this a little later on, but this is just a quick overview. Blue mRNA is going to copy the DNA. It's going to copy the gene that it wants to make, not the whole DNA. And we're going to talk about this later, but the gene it wants to make, and then it leaves the nucleus and goes out to the cytoplasm. And it's then going to attach to a ribosome. And remember, the ribosome is made out of rRNA. So this little green guy here is rRNA. And it's going to literally read it like it's reading a code. And it's going to attach to it. And it's going to read it every 3 bases. Because remember, 3 bases is a codon. And every 3 bases is going to mean a specific amino acid. So now our final player here is our tRNA. So tRNA has 3 bases on it, which correspond to this. So that's how it knows what to get. And it goes out and it picks up an amino acid. So it's going to pick up that amino acid, it's going to attach to it, and it's going to carry that amino acid to the ribosome. And it's going to attach to it. And it's going to then drop off that ribosome, and it's going to leave behind its amino acid. And then that's how the whole process gets done. So these tRNAs are going to bring the amino acids to the ribosome, and you're going to build your protein. So the 3 types of RNA are mRNA, which carries the message to the ribosomes, rRNA, which is the ribosome, and tRNA, which goes and picks up the amino acids from the cytoplasm and brings them to the rRNA. Thank you very much.